few people may still just trickle down, and we'll just go ahead and get started. So it's a real pleasure to introduce Adam Lowering, who's our speaker today. And Adam received his PhD in molecular biology in 2000 at the University of Washington, and he also received his medical degree also from the University of Washington. And during his graduate studies, he focused on retroviral pathogenesis using feline leukemia virus model. Then he continued his postgraduate work at UC San Francisco, and there he focused on the evolutionary dynamics of poliovirus, and also completed his medical training. In 2012, he came to U of M and joined the faculty at the medical school. And today he's going to talk to us about tools for studying RNA virus diversity. Thank you.
Thanks. Can you hear me okay? That's probably good. All right. Well, thanks. This is fun. I've met some of you along the way. I hope to meet more of you. I consider myself kind of bioinformatics light. I'm your end user, but because of some of the questions that we've been asking, we've had to make some forays into your turf a bit. And so I'll tell you a little bit about that today. I hope to get some input. So my lab, my research interest is in RNA viruses, particularly their evolution. RNA viruses as a group are remarkably successful pathogens. They cause a wide range of illnesses, from respiratory viruses, the common cold, flu, infectious diarrhea, encephalitis, hemorrhagic fevers, which are always in the news, as well as hepatitis, and then many childhood rash illnesses. One of the reasons they're such successful pathogens, or at least, yeah, one of the reasons is that they're very adept at evolving. And this allows them to do a number of things. In the case of flu, be a zoonotic and other zoonotic agents, which can migrate between multiple species. There are a number of RNA viruses that are arboviruses or insect vector viruses that can replicate in species of mosquitoes and humans or primates separated by millions of years of evolution. They're quite good at escaping immune surveillance. One of the reasons, of course, why we have to get a flu shot every year. You should get yours in the next few weeks. And so escaping the antibody or CTL responses. And then finally, drug resistance. Often the first indication of a drug target for a virus is isolating the resistance mutation, because it can evolve quite readily. And generally accepted that RNA viruses evolve so rapidly to a large degree, but not exclusively, because they have very high mutation rates. This is a diagram which I think illustrates it nicely, which shows the mutation rate versus genome size. And so most of us, or all of us, are down here. And then RNA viruses have mutation rates that are several orders of magnitude higher than DNA viruses or DNA-based life forms. And that's because with one exception, the RNA-dependent RNA polymerase that copies the viral genome has no proofreading or repair activities. And so therefore, the virus is unable to correct its mistakes. The polymerase is unable to correct its mistake. And the mutation rates for RNA viruses range between 10 to the minus 4 to 10 to the minus 6 mutations per nucleotide copy per cellular infection cycle. Most RNA viruses are roughly 10 kb long, their genomes, and so that translates to approximately 1 mutation per genome replicated. And so every newly generated genome, in theory, is distinct from the parent genome. And this means that the virus could quickly become a population of closely related genetic variants. Okay? This population is called a cloud, a swarm, a quasi-species, genetically diverse population. Any term you want. I like swarm, although cloud is nice too. And there's a large amount of interest in understanding what this diversity is there for and what it means. You know, is having this sort of swarm different from this sort of swarm, basically, and does that affect how the virus behaves? And does that explain differences between either different viral species or different variants within the same species? And that's a question that we're very interested in. And we approached this so far with 2 viruses, poliovirus, in which I did most of my postdoctoral work. Poliovirus being a very nice model system. Of course, it's still a virus that causes disease in people, but a lot of its power now lies in the ability to do things in the laboratory quite easily with poliovirus for a number of reasons. It's also a well-studied virus, so a lot of the molecular biology is well worked out, and so and the genetics are quite easy. But then more recently, we've started to branch out into influenza virus, and those projects are a little bit more translational, where we're looking at influenza viruses in people. And I'll tell you one project in each today. One about an informatic tool we've developed with the help of Ryan Mills in your department for manipulating genetic diversity. And the other is a tool that we are looking at in influenza for characterizing or defining diversity. And so those are the 2 things we're going to do. And so first I'll tell you about manipulating diversity. And really it's like, well, diversity is there, but to really study what it's doing, you'd like to have some way of controlling it. You know, having diverse swarm A or diverse swarm B, and then, you know, distinguishing what's different. And that's a problem I started to look at in my postdoctoral work and continued here. And one way I get at this is, I told you that the virus has a high mutation rate, and that's kind of the engine that generates genetic diversity, right? However, there's a lot more to mutation rate in determining patterns of diversity in a population. And this slide, I think, illustrates it well, is that you have HIV and influenza have essentially the same biochemical mutation rate. They make errors at the same frequency. However, this is a tree showing sequences of HIV in the Congo in 1997. And then this is a tree showing influenza, still a pretty diverse virus, worldwide in 1996. And so you can just say, well, you know, one virus has a lot more diversity that it can express than another. And so there's more to it. And we've been interested in understanding mutational tolerance of a virus, also known as mutational robustness. And the idea is the virus mutates, and you get, you know, a dominant variant, but then all these other little variants around it that are related. And which ones are there depend on how well that mutation is tolerated. Most mutations are bad, and probably about 30 or 40% are lethal to the virus. And so you can make that mutation, but it's a dead end. And it's never going to rise in frequency in the population because the virus can't replicate anymore. Many more mutations are detrimental, so they can't increase in frequency because they're essentially a crippled virus and they can't replicate well. And so a virus that has, in general, mutations that don't cause as much damage will be tolerant of mutation or mutationally robust. And the idea is that a more robust population can maintain a larger degree of genetic diversity. Okay? And that might be another thing that controls patterns of diversity in a population. And so, well, gee, that's a neat idea. It's been around for a long time. There are a number of things that probably affect how well a virus tolerates mutation. And one question I became interested in is, does codon usage by the virus affect mutational tolerance? And I'll walk it through with 2 different schematics. The first one is here. And the idea is that, you know, how you use your codons determines how well you can buffer mutations in your genome. And excuse me. So here's an extreme case of arginine, which is encoded by 6 different codons. And so in the protein, it doesn't make a difference. It's arginine. It has the same charge. It does the same thing in a protein. However, different arginine codons mutate non-synonymously at different rates due to the connectivity of the genetic code. And then within the non-synonymous mutations, you can get different. If you start with an AGG, you can get these different amino acids as their one mutant neighbors. Whereas if you have these, you get very different neighbors. And so the idea is maybe codon usage doesn't affect the protein, but the one mutant neighbors will give rise to very different proteins. And at in a virus that has a high mutation rate, maybe this would have an effect on overall, the virus's tolerance of mutation. And that's the basic idea. And so walking it through, another example is you have this Elvis peptide that's encoded by two different stretches of nucleic acid. Okay? So different codons, but they still code for Elvis. However, the one mutant neighbors of all of these are different. So if you start with this nucleic acid, when that Elvis peptide gets mutated, it's going to be a different peptide than if this Elvis peptide mutates. And of course, these will be located in different regions of sequence space, whether Tennessee or Nevada. And of course, these would give rise to Elvises of very different phenotypes. And so the idea was basically, can you generate viruses that have large-scale synonymous mutation across the genome? So they start with the same amino acid sequence, but they start in different positions in sequence space and are going to mutate differently, and thereby create different populations. Poliovirus, one of the nice things is it's the first virus that has been chemically synthesized using long oligos. And there were several viruses already generated called MAX and SD, which have either 566 or 934 point mutations across the capsid region of the genome. And so they code for the same capsid protein, but they're going to mutate differently because their primary nucleic acid sequence is different. And so we're going to compare these two synthetic viruses with large-scale synonymous mutation to the wild-type virus in our work. We were able to show that, indeed, using deep sequencing, that these viruses did give rise to a different mutational spectrum. Okay? Shown here are the codons across the capsid. It's 881 codons. And this is basically a heat map where each little bar is a mutation that was detected. And the scoring, the heat, is the Blossom 62 score, which is empiric measurement of how common a mutation is, and it's used in, I don't need to tell you, in alignment algorithms. And then across the top is this trace, which shows how variable that region of the capsid is, looking at all sequences, in GenBank. And so this, the capsid is a tightly organized structure, but there's also loops that are more variable than others, and then there's more conserved protein-protein interfaces that are less variable. And I think you can just quick glance and see that wild type and then the two synthetic viruses, MAX and SD, had very different patterns of mutation, and they gave rise to proteins with very different amino acid substitutions. And so by switching the nucleic acid, we indeed give rise to populations that have different functional variants. And then we went further to show that basically by doing this, we made the virus more or less tolerant of mutation. And so this is an assay where we treat viruses with ribavirin in culture, and ribavirin is a mutagen. And so it gets misincorporated and templates G to A and C to U mutations. And so in a dose-dependent manner, we can increase the virus's mutation rate. And then if a virus is less tolerant of mutation, it will lose titer. And we found that SD was actually hypersensitive to mutation. So it was less tolerant of these newly generated mutations, and that would be consistent with our model in which codon usage functions as a buffer for mutational load. And then, surprisingly, when we put these viruses into mice, we found that mutational tolerance was actually important, in that the most sensitive virus to mutation, SD, was also attenuated in mice. And so that implied to us that viruses need to be able to buffer their mutational load in order to maintain a stable population, and that is somehow important for how they behave in vivo. Okay. So this work was published in 2012, right when I came here, and took a while to publish, I'll tell you that. And we got a lot of reviews, and everyone raised very important caveats. And one is, of course, low N. We did this with 3 viruses. That's cool, but that's not a lot. And then probably more relevant for this talk is that, well, yeah, you did these synonymous mutation, but synonymous mutation will affect a lot of things, not just mutational tolerance, and have you adequately controlled for these factors? And we did, but you can never be sure. One, you change the sequence. These viruses have a lot of RNA structure in their genomes, and so by changing the codons around, you might be disrupting important RNA structures. You may be affecting codon pair bias, which may or may not affect gene expression. You might be affecting dinucleotide bias. Certain dinucleotides are thought to be sensed as like PAMPs, as pattern signals that the immune system recognizes. So, for example, CpG is sensed by toll-like receptors, and maybe you're altering that. Maybe you're introducing spurious microRNA binding sites, and so we're like, okay, well, there's a lot of things to control for. It sure would be nice to control for all these things and isolate mutational tolerance as one factor. Unfortunately, we thought, well, you should be able to do that, right? Because synonymous sequence space is large. If you have a 20 amino acid peptide that's coded by one of each of the 20 amino acids, back of the envelope calculations, you can have 10 to the 18th different primary nucleic acid sequences that will give rise to that same peptide. So that's a lot of possibilities that you can try, and you should be able to find something that controls for all these other things. And so soon after I came here, I sent an email. I don't remember who I sent it to, but somehow I wound up getting to Ryan Mills, who has helped us enact this project. And this is all the work of Daniel, who's a postdoc in my lab, who has an interest in informatics. And we developed this tool called Codon Shuffle that I'll tell you about, that's implemented in Python. And so it starts with the idea of, well, we wanted to generate sequences that we could synthesize and test. And we do this using a variety of different permutations, algorithms, and basically swapping either single nucleotides or dinucleotides or trinucleotides, essentially keeping the amino acid sequence the same. And then just that, by shuffling them around your sequence, you generate a lot of different primary nucleic acid sequences. So it's just basic, permuting the sequence, but keeping the coding sequence the same. And then the second step is analyzing the sequences. And, I don't think we came up with any of these metrics on our own. We basically used existing tools. So, for example, you can measure the codon bias via the codon adaptation index or the effective number of codons. We used both M-fold and Vienna RNA to measure the free energy of RNA folding across our sequence. We measured dinucleotide bias. We measured codon pair bias, et cetera. And what's shown here is we permuted the poliovirus sequence 1,000 times. So we generated 1,000 different permuted sequence for each of these 4 permutation algorithms, so about 4,000 sequences. And then we generated a histogram of how these sequences, their values for a given, whether it's CAI or the free energy of RNA folding. And then we're like, okay, well, what does that mean? Nice, you can generate a histogram. How is the codon adaptive index of 0.675 really that different from one of 0.7 for poliovirus? We didn't know. And so what we plot along with this is the CAI for, or the folding for other enteroviruses. So we had a panel of 89 enteroviruses sequences from GenBank, and we did the same metric. And enteroviruses all have the same general structure as poliovirus. They have the same replication strategy. So it's sort of a control set. These all replicate in people, and so these are ones where presumably over this range of free energy, that's fine. It's kind of a biological control for what we're doing. And so we did this for 4,000 sequences generated these distributions. And then the goal was, well, we want to pick sequences that are as similar as possible to wild type, right? So they have the same CAI as wild type, which is highlighted as this dotted line, as close as we can. The same free energy of folding, the same dinucleotide bias, et cetera. And here's where the real challenge came is, well, how do you do that? Particularly when you've got all these different metrics that have different scales and different raw values. Like, how do you say something's 0.7 versus 0.725 versus minus 800 versus minus 600? And actually, I'll come back to that. So what we did is essentially we have these normal distributions. We know where wild type is. And essentially we said, OK, how far by z-score are these from the wild type? And so something that's close to wild type, you know, the z-score should be low, the delta z should be low. Something far, the delta z-score should be high. So how many standard deviations is it off? And the advantage of this is it kind of normalizes across scales and values into a single metric that you can use. And what we did is then did a least squares regression to essentially say, okay, across codon pair bias, folding free energy, effective number of codons, codon adaptive index, and dinucleotide frequency, which of these 4,000 sequences is the closest by delta z across all those delta z scores, okay? And we call this its least squares distance, or D, which we plot a least squares distance versus the Hamming distance in nucleotides across all our species. And here, for 1,000 sequences permuted with this DN231 algorithm, we have all these dots, each represent a sequence, and you can see that there's a number of them that have a least squares of 0. So on average, they are pretty close to wild type in all these other metrics. And yet they still differ by 550 to 600 bases. So quite distant, genetically at the nucleic acid level, identical at the protein level, but yet all these other factors are generally averaged out as being similar to wild type. Okay. So then we were like, well, how well does this perform? And I'm not talking about performance in the informatic literature in terms of how many CPUs and how quickly it runs. I'm talking about, well, how good are we exploring possibilities? And so what we did is we ran it 10 times. So we generated 10,000 sequences. And we're saying, are we hitting on the same solutions? Are there only so many solutions to this problem to get a sequence similar to wild type? And indeed, there's a lot of solutions. So this is just a haplotype accumulation curve where we measured the number of sequences versus the number of unique haplotypes. Every haplotype was unique. So we ran it 10,000 times. We got 10,000 unique sequences. Okay. And then this is plotted just the same plot, only each color is a different run of 1,000. Okay. And so again, least squares versus Hamming distance. Here are the ones that are closest to wild type with the least squares of 0, and they're all distinct sequences. So you could hit on something that's very similar to wild type across this range of metrics, but with very different sequences. And then this is just a neighbor joining tree showing these, I think, forget how many sequences there were here, 80, 90, something like that, that have this least squares of 0. They all differ from each other by about 500 bases. So it's not like there's this, single ancestor that they're all similar to. They're all as different to each of the other ones, which is consistent with what I told you about synonymous sequence space being really large, and there's a lot of possible solutions to this problem. And so this was really exciting because it means, well, gee, you can do this. You can permute the sequence to your heart's content and still get out your desired product and a lot of candidates that you can try. So we just published this, again, with a lot of heavy lifting and help from Ryan. And we think there's a number of applications for this tool. So one is you could use it to choose sequences that are similar to wild type in every way and ask, are they more or less mutationally robust? And that, of course, was what got us into this in the first place. So that's one thing we're interested in. How about you could also say, well, we could choose sequences that are different in every way. Okay, you could say, I want to pick a sequence that's over here, that's different in its codon pair bias and its dinucleotide bias and everything. It's just completely different from wild type. And you're like, well, why would you want to do that? Well, there's been a number of papers where synonymous, synthesized synonymous altered viruses have been used in vaccine design. Initially in polio and now in flu, VSV, dengue. And the idea is you're pushing the virus in a way, you're altering its genome, and you're taking a sledgehammer to its genome and changing the way it replicates because you're altering all these things that are probably important for replication and gene expression. But yet, the little bit of the virus it makes, it's going to have the same amino acid sequence, right? So it's going to be immunologically identical as recognized by your immune system. So you can attenuate the virus, preserve its antigenicity, and be a live attenuated virus. And so basically, we might be able to do this by altering all these things to make more complete attenuation and make it less likely that the virus will revert to virulence. And then you could just try to understand other factors. Maybe you just want to look at CpG bias. You could generate a virus that has everything else the same, but it's just different in CpG bias and understand how CpG bias affects the virus's behavior. And so we think this is a tool that'll make people in my field able to better answer some of these questions in basic virology. And in a little bit of a venture, we're now on GitHub. And so we have it there, and we just got our first pull request, that someone's been making some improvements to our software. So that's kind of fun, because it means people are using it. So that's the codon shuffle work. And so now I'm going to move into our flu work, where instead of manipulating viral diversity, we want to understand natural viral diversity. And in cell culture, it's easy to generate viruses with a lot of diversity, but in people with a viral infection, there's probably not the same amount of diversity. And another question that's very interesting to us is, what happens when a virus goes from one person to another? In a sneeze or touching something and putting on your face or whatever happens, how much of that diversity gets transmitted from one person to another? And what can you understand about infection, vaccination, by understanding how diversity changes in natural infections as they're transmitted? So we've been working a lot to develop both the bench protocols and also an informatic pipeline for deep sequencing of viral populations directed against getting samples from individuals. And so our sample that we're going to be using in the flu studies are essentially a throat swab of someone. So take a swab of someone's throat, put it in media, and then we take that, do an RNA prep, develop a sequencing library, and characterize the diversity. So very different from taking a cell culture dish with tons of virus in it and looking at diversity. And so we've optimized a multiplex RT-PCR to simultaneously amplify all 8 genomic segments of the flu genome. Flu has 8 chromosomes. And so we can do this quite well now. We get very nice sequence, and we sequence to very high depth of coverage, about 20,000x, with the idea that a well-covered diverse library would allow you to call rare single nucleotide variants in the population, which is our ultimate goal. And then where we've been spending a lot of time is on the informatics, and this is largely the work of JT McCrone, a graduate student in the lab. You get all pixelated at this size, JT. So I don't think I need to tell this audience that there's a lot of sources of error in next generation sequencing. And this is from a review from George Church that characterizes some of them. And while there's sample preparation, in our case, that's a biggie, because we start with an RNA genome, so we always have to do a reverse transcription before we do a PCR. Reverse transcriptase has a high error rate, and so that's something that we can't get around. And so that's an important source of error that is probably more specific to what we do than, for example, a human genome where you can sequence at 40x coverage and get everything you need to know. And of course, ours is not on a simple diploid setup. Library preparation is another issue. And then where there's been a lot of attention is the sequencing and the imaging and calling a base. And of course, when you're calling a base 20,000 times, you're just going to have, a low frequency error can be hard to distinguish from a rare variant. And that's our basic problem. And so I distill it to how to identify a mutation. And as a virologist, it's like, well, if you look at a gel and you see a band or no band, that's simple, right? And if you're a little more modern and you've got a chromatogram and you've got a red and black here, there are ways you can just look at that and tell, well, that's probably something we're not going to be able to call. But if the red were a little higher, we'd have more confidence that maybe that's, that base. But then the problem is, of course, when you get 20,000 little peaks like that, how do you do it? And so I think the issue for us is specificity. I think the field, at least in my field, everyone's focused more on sensitivity. Can you detect these rare variants, right? Does your variant caller find things that are 1% or less? There's a tremendous amount of focus on that, but specificity, I think, is a bigger problem. And that's illustrated here. I won't tell you what paper it came from, but essentially, here you have intrahost single nucleotide variant frequencies across a range of samples. There's a star here, so there's a significant difference, right? But you can see, well, gee, really, okay, these and these, yeah, they sort of look different, but these are all pretty rare. And if this is what you're measuring, you really need to be sure that these things are all real and not, noise. You might just be finding slight differences in noise that achieve statistical significance. And so what is often reported in the viral literature is like, well, we achieve 99% specificity with our variant calling, okay? Which sounds great, right? 99% is awesome. But if you have a 10 kb genome with 3 possible bases at each position, there's essentially 30,000 possible variants that you're calling. So if you have 99% specificity, you have 150 false positives with 99% specificity, which is really high. I mean, that's almost all your signal. And so we think you need to get a lot higher than 99%. And our goal was 99.9% specificity, which wouldn't get you to 0 false positives, it would get you closer. So there are a cornucopia of variant callers out there. Here's just a few. So we didn't develop a variant caller, but we want to make them better, the ones that other people have developed. And I think a real issue is, my little hobby horse here is in the informatic literature, a lot of these variant callers are benchmarked with simulated reads or things like that. So we simulated a bunch of reads, and then we look, oh, we can find all the mutations, and our specificity is 99%. Well, that's very different for the end user, right? Who's like, okay, I'm going to start with a bunch of RNA, and then I'm going to use your variant caller, and I want to believe what comes out. And because there's biological sources of error that the variant caller is not going to be able to control for. And then there's certain issues, as I'm sure many of you realize, that are specific to your experimental setup. And so one variant caller might not meet all needs. We settled on one, which is called DeepSNV, which was initially developed to look at tumor heterogeneity, so calling SNPs in tumors. And what it essentially does is it uses a binomial model to detect false positives from a real variant. And the idea is you sequence a plasmid control in the same flow cell as your experimental sample. So you take a plasmid, you put it through the same library prep, you sequence it in the same flow cell, and so you have a large number of reads on that plasmid sample. And that'll tell you kind of the background noise. And a true mutation in your sample, shown here as red, will have a different frequency profile. And then you can distinguish them using a binomial. And why we chose this variant color out of all of them is it's one of the only ones that we saw benchmarked with actual experimental data. So, you know, I hope I'm not slamming simulated reads because there's a lot of value, but they actually took, you know, PCR products and then mixed them and then showed they could detect. And that made me feel kind of warm and fuzzy. So that's why we picked it. Nothing else special about this variant color. And then we designed an experiment because that's what we do. And you'd be surprised that I don't know if this has ever been done in the virology literature for all the deep sequencing that's been done and published. I don't think I can think of a paper where someone's actually said, okay, we've benchmarked this and we know we can find a mutation at this frequency and what our false positives are. And so what we did is we have 20 viruses, each with a single nucleotide mutation, okay? And each of those viral stocks has been Illumina sequenced, okay? So we know that it has the mutation, and we know if there's any other kind of mutations that have arisen in that viral stock. Because remember, the virus is going to make its own errors. We diluted these. We also have a wild type sequence that has been Illumina sequenced twice, okay? So we know what the real variants are in our wild type population. We diluted a mixture of these 20 mutants into the wild type at varying frequencies, from 5% of each mutant down to 0.1% of each mutant. And then we varied the input by titer. So we either start with 10 to the 5th viruses all the way down to 100 viruses, okay? And so we've got 20 different samples. We made duplicate libraries of each and put them all in one HiSeq run, okay? And so we have multiple levels of control that detect true and false positives. That's basically what I want to get across. And then we say, okay, we've been using ROCs to say, okay, how do we do sensitivity, specificity? And here's where we started. Our viral titer 10 to the 4 down to 10. Frequency of the mutations, basic color. And basically, all I want to get across here is we did pretty well, right? Using off-the-shelf default parameters on the variant color, we can get down to, certainly greater than 99.5% specificity. And we start losing sensitivity at lower variants. And then also, of course, when you increase your specificity, you're going to lose your sensitivity. And that's the trade-off that we're looking at. And so we're like, okay, well, this is good, but can we do better? And we started playing around with a number of things and chasing down, because we know what our false positives are, because we know what went into the mixture, and we know we're trying to find 20 mutations, and there shouldn't be anything else, okay? And so we're like, okay, what are we finding that's not those 20 mutations, and what characteristics about them can we figure out? One, just to show an example here, is, well, if this is plotting the position of the mutation on a 100 base, HiSeq, read 2 by 100, and saying, okay, where is the mutation call on the read on average? And remember, we have high depth of sequence, so we have a lot of reads that call a mutation. And basically, the false positives are shown here, and they tend to be towards the end of the read, even with a good quality score. So your quality score is greater than 30, but they're on the end of the read, and they're false positives. Whereas the true positives, the average read position of that mutation is in the middle. And so we can just apply that simple cutoff to get rid of a lot of false positives. So that's just one example of how we zeroed in on which things are true and which things are false and how to filter them out. And it's all empiric. So then, let's see here. The way we do full screen. JT set up this, it's like a PI's dream, is the Shiny app, which I assume many of you use. I love it because JT can load up all the data and I can just play around with it and see. So it's little work on my part, but a lot of information, and so you can just play around with data, and it's fun. And we ultimately hope to publish this soon and put this on GitHub, let people play around with the data, and even put our reads available. So if someone wants to benchmark their variant caller, they can do that. Like they don't have to do what we did, but they can see if their variant caller is good or not. And so what we do is we have all these different variables. Shiny. Oh, you know what? Okay. So I think I put the advanced. So we get the ROCs curves. We can vary our p-value, cutoff, whether we do duplicate reads or not. You know, what kind of binomial model we apply, the read position cutoff at the end, all those. And then we get a nice table, and we can see how many false positives, how many true positives. I'm not going to clarify it again. And benchmark ourselves. So kind of where we got our sweet spot is with the two-sided beta binomial, the read position cutoff, and then one thing is we don't call anything less than 0.4%, so 1 in 250. So that's kind of as low as we can go. And you can see, you know, the ROC is a vertical line. And so we can get down, and you can see from the table, you know, in our false positive column, we get essentially close to 0 false positives, and we get pretty good sensitivity. So this is kind of where we stop. And exit, get back to PowerPoint. Okay. So then the last problem was virus input, which is not an informatic problem, but it's another insurmountable problem for us, right? Because I remember we're getting a swab out of someone's throat, and if we're lucky, there's a lot of virus in there. If we're not lucky, there's not. And if you think about it, if you start with, you know, 10 to the 2 genomes per microliter versus 10 to the 6 genomes per microliter, you're going to have to go through more PCR cycles, right, in making your library. And that has the potential to introduce false positives that you can't get around informatically. We could try to zero in on those and apply more stringent criteria, but then our sensitivity goes in the toilet. And so we know from our ROCs that at 10 to the 5th or above, we can get 0 false positives. So in a lot of our measurements, we're going to be focusing on samples that have higher amounts of virus in them. And we're going to be less certain about samples that have lower amounts of virus in them. And that's just shown here. So 10 to the 5th copies in a mixing study versus 10 to the 2 copies in a mixing study. And that, so before it was tighter, now it's genome copies by qPCR. And then here's the specificity again. So at 10 to the 5th copies, we get, you know, perfection in terms of our specificity. But as you get more amplifications in your PCR, you start getting more false positives. And then, of course, your sensitivity is going down because you're starting with less. So your chances of finding a rarer variant are less. And that's just something we can't do anything about. But the important thing is we understand it. And we know where the limits of what we can say are when we're looking at real human sample data. So we are in the process of kind of putting this together and publishing it kind of as a standalone. You know, this is a benchmarking study of a variant caller, and we're actually doing a couple of variant callers and saying, look, the viral community, you need to pay attention to this because it becomes kind of a data reproducibility and believability issue. And you know, because there's a lot of studies of diversity, but we don't know because none of these things have been benchmarked well whether the data is meaningful or not, or whether people are making inferences based on noise. And so we're going to try to make it all publicly available, put the reads up. People can benchmark their own pipelines using our data because it's a well kind of curated data set. And so just finally, I'll finish up with just a little bit of what we're doing with this variant caller. One thing we're interested in is how does vaccination impact viral diversity? And we're doing this in collaboration with Arnold Monto and Suzanne Omid at the School of Public Health. And they actually ran the last placebo-controlled randomized trial of influenza vaccine. You can't do it anymore because universal vaccination is recommended, so everyone's supposed to get a flu shot, and you can't have people getting a placebo flu shot anymore. But in 2006, 2004 through 2008, it wasn't, and so they ran a large trial with 1,600 people in the vaccine arm, 300 in the placebo arm, and saw who got influenza. And these were generally University of Michigan students, so they were fairly young, healthy. They didn't have a vaccination history because it wasn't recommended to get a flu shot, and 20-year-olds don't get flu shots anyway, right? And so, there's no prior vaccination to mess things up. So it's as clean a data set as you're going to get. And so what Kari is doing in the lab is, the flu vaccine works 60 to 70% of the time. Its efficacy. So a large number of people get the flu shot, still get the flu. Okay, still get your flu shot, but don't expect miracles. And basically, do the people who got the flu shot but still got the flu get different viruses than the people who got the placebo? And so what we've done is we've gotten these swabs out of the freezer and Illumina sequenced them and applied our variant caller. And here's just preliminary data, just a few things we're working on. So what we're looking at is by day of reported symptoms across the whole genome, how many single nucleotide variants are we detecting in both the vaccinated arm and the placebo arm? And then we're looking at a gene level. So you might think in HA or NA, which are the surface proteins of the virus, where your immune system targets, there might be differences whether you get the vaccine or not. So we're looking at the data for that. And then we're also looking for, well, are these different viruses, depending on whether you're in a vaccinated host or an unvaccinated host, exploring different solutions, right? And so maybe all the ones in the face of partial antibody response are evolving a certain way and sharing certain single nucleotide variants. And what we've noticed is there are some single nucleotide variants that are in most of our samples. So they are common, probably what was circulating in Ann Arbor that year relative to the reference. But then there's a number of them that are not quite private mutations. They're not just in one sample, but they're in a few samples. And so that's something that we're going to be exploring further. And then that brings me to another project, which JT is working on, is, well, if there's a certain number of variants that might be shared among a couple people, can you use that to start looking at what's transmitted from one person to another, right? And the idea is this. I just had a cold. My 4-year-old and 6-year-old had colds before me, right? So they gave it to me, right? Right? It's the kids, and I don't know how many of your parents, but that's the constant refrain. Oh, my kids started going to school, and now I got sick all the time. Is that fair? I don't know. Could I have gotten sick from someone at work, and it's just coincidence? And so it would be nice to know, can you use the sequence data to link people in transmission chains? And essentially say, yes, by using the sequence data and performing inference, yes, it's highly likely that your kid gave it to you, or no, let's let the kid off the hook. And so JT is working on this Hive cohort, which again is run by Arnold Monto at the School of Public Health. There's about 300 families of 4 or more in southeastern Michigan every year that they follow. So about 1,000, 1,500 people. They just follow them through the year. Anytime someone gets sick, they get swabbed. So there's a lot of rhinoviruses, coronaviruses, but then there's also influenza viruses. And then about 50 or 70 a year, you get 2 people in the same household within a couple days of each other who are sick. And so what we're doing is we're sequencing all those viruses in household transmission, putative transmission cases, and across the cohort as a whole. And then we're going to look for shared variants and essentially say, well, essentially use that to make a distance matrix and say, well, if you share a number of these variants together, it's highly likely that you're linked as a transmission chain as opposed to, someone working at the bank, the day you went there. And so trying to get a sense of, how confident are we that this household transmission event happened? Because then we're like, well, if we know a transmission event happened, then we know we can start defining what's going between one person and another. And then we know some of these people are vaccinated, so we can say, with vaccination, is there a titer bottleneck? Do things change more? And try to understand that transmission process and what we can do about it. So we're at the early stages, but it all has to start with informatics that you're confident in, which is why we spent so much time on our sequencing pipeline, because if we're going to do a secondary analysis of the data, you have to be sure that your primary data is solid. So to summarize the influenza part of the work, reliable detection of intra-host single nucleotide variants is tricky. No matter what you read in a paper, it's tricky, and ask questions. And what works on a variant caller for human genomes or bacterial genomes is not the same as what works in a variant caller for an RNA virus. It's even trickier to do this in clinical samples, where you may have to do a lot more RT-PCR to generate your sequencing library than in a cell culture derived sample. We developed what we think is a robust pipeline. And like I said, I think one of the big things that we're going to be able to do is give this data set out to the community, and people can, well, I don't like your pipeline, I'm going to do a different pipeline. They've got a data set that they can say, okay, we know how many true positives are, we know where the false positives are, and then we can optimize our pipeline just like we did. And then we're using this to move forward with studies of intra-host diversity, which are ongoing, and hopefully in a couple of years I'll be able to tell you what we found. So I'll thank my lab, many of whom are right there. I talked about Daniel's work on Codon Shuffle, JT on the pipeline, and a little bit about Kari's work with the flu vac samples. We have other interesting stuff going on that other people are working on. Arnold Monto and Suzanne Omid are collaborators on the flu stuff at the School of Public Health. Ryan in your department, who we wouldn't have been able to do Codon Shuffle without him, and he's been really fun to work with. We got started with kind of the framework of our pipeline with help from Chris and Peter at the Bioinformatics core, and then our funding sources. So I'll take any questions. Yeah, or maintain predicted function, right? So change the primary nucleic acid sequence without altering things that might affect the overall function of the virus. Yeah.
So, you know, it just may be something to look at where you can copy and paste, but there's kind of a whole area of protein design where they do something very similar. You may be able to copy and paste some ideas and see if they improve your results.
Yeah. Yeah. You mean in terms of like methodologic? Because of course we're trying to keep the protein sequence the same.
So you're just changing?
Just changing the nucleic acid sequence.
Oh, okay.
Yeah. Yeah. Yeah, because we want to keep the amino acid sequence the same. Yeah. Good. Yeah, thank you. So the question is, do we look at indels in our benchmarking? And we just go like this with indels, because they're probably not as biologically common or relevant as in many other genomes, right? Human genomes in particular, right? Because a lot of the viruses, it's like, most of what it is, it's like an open reading frame. You throw an indel in there, it's essentially gone, and they're probably pretty rare. And there's even more issues with detecting indels. So we just said, we're agnostic about indels, and we're not going to study them at all. And maybe we're missing something, but I think JT would kill me if I told him to improve indel detection. Yeah. Go.
Very nice work.
Thanks.
Early in the lecture, you drew a contrast between HIV virus and poliovirus.
Yeah.
Of course, they're totally different molecular structures, but from the variables that you study, what are you able to correlate or explain in their evolution and their function?
Yeah, so it's not nothing. How's that? Nothing. All the money spent, we haven't found anything. I think the answer is there's probably a lot of things. And so there's been a number of studies where people, correlate various things, evolution, because basically it's a problem of mutation rate and evolutionary rate, right? So HIV has a much higher evolutionary rate than poliovirus in terms of substitutions per site per year, right? It evolves. But the mutation rates are the same, and there's a lot of things that go in: bottleneck size, demographics. How many people are in the world are infected with poliovirus versus HIV? The host size, demographics. Like we saw this in the Ebola outbreak, right? The initial estimates of the substitution rate were vastly different than later in the epidemic, and it probably had to do with kind of the explosive, burst when the epidemic started versus when it tapered off. And then on the kind of the basic nuts and bolts of the virus, I think there's probably a lot of things. I think mutational tolerance is one of them, but even there's probably variation in mutation rates across a genome, right? That maybe the envelope sequence of HIV has a different mutation rate than an internal protein, and maybe the virus is able to has more loops and is more tolerant of mutation in loops, and how the proteins are structured is probably affecting it too. So I think I doubt. I mean, I presented it as mutational tolerance is going to be everything, but I bet each virus is going to have like 5 different things that build on its mutation rate and affect how it evolves. Does that answer your question a little bit?
So I think you showed that the false positive variants tend to distribute towards the end of the sequence, but there is a small bump in the middle too. Can you explain why there is a small bump?
No. So the ones that'll be in the middle, right, will be, so the point I was trying to make, I glossed over it with kind of the ends, because probably this audience knows that a quality score of 30 does not mean a quality score of 30, right? That it doesn't mean that there's 1 in 1,000 chance that there's a mutation there. And certainly our data show that a quality score of 30 on an end of the read does not mean the same thing as a quality score of 30 in the middle of a read, right? Because we had Q30 base calls on the end that were false, right? And so that was an important thing. But then to your question specifically, it's like, well, in the middle, those are high quality bases, high map quality scores. They're like, they look real, right? And we're calling them false positives because they're not expected, right? Because we know 20 true positives, and we're calling everything else false. They could be RT-PCR errors. They could be that 1 in 1,000 with a quality score of 30, that happens enough times that we can detect it. And I think it's kind of like the viral Heisenberg uncertainty principle. At some point, you just can't get any closer to certainty that something's real or not. And so we're calling it a false positive. It might be a true positive. And so our goal has basically just been to get rid of as many false positives as we can, and some we're just going to have to say, we're pretty sure they're real, and leave it at that. Is that? Yeah. But there's no informatics signal that we're like, okay, those false positives in the middle have this characteristic, because if we knew that, we would have figured a way to filter them out. Does that answer?
In your the influenza vaccine versus the placebo effect, and you didn't dwell on the results, but I think did you show when you looked at hemagglutinin that the diversity was larger in the in the placebo group?
Yeah, this is largely playing around with data, and I glossed over it a little bit. So because we've improved our variant calling pipeline, we've improved our primary data analysis since the secondary data analysis, and we haven't redone the secondary data. So this is kind of teaser. We did find that there was more diversity on day 4, I think it was, in hemagglutinin in vaccinated versus placebo. But we need to go back now with what we've learned in the last 3 months and redo the secondary analysis because we've gotten better at the primary analysis. So it could be that some of this signal goes away, which is exactly the point we're trying to make to the community. It's like primary data has got to be really good to do this sort of work. But those are the questions we're interested in because we're like, well, gee, that would be kind of cool, right? If vaccination affects the diversity on hemagglutinin, that would be, you know, something we'd be excited about. Cool. Yeah, thank you.